So next up, we have postdoctoral fellow Jay Thangapan. He is from Dr. Sylvia Ruskin's group in the Whitehead. He grew up in a French colonial city in the southern part of India known as Pondicherry. He did his bachelor's and master's at Pondicherry University in biotechnology and bioinformatics, respectively. He next moved to South Korea for grad school, where he obtained a PhD in chemical engineering with a specialization in protein engineering at Busan National University. Jay says his favorite thing about the Earth is the opportunity to travel, explore, learn, and challenge his views. Whenever he looks at the sky, it reminds him that there is so much more to experience and learn, both in biology and in life. So please join me in welcoming Jay.
Hello, everyone. Can you imagine structure can control biology? I would like to introduce the concept how structure can play a big role in the whole biological process. If you see genes are the recipe for building any proteins. From the traditional view of biology, we think the sequence it's all about. It's not just the sequence, how it is arranged and folded makes a huge difference. So the traditional view says that DNA has all genetic instruction required for a life. A DNA copies RNA, all this genetic information, but RNA has the responsibility to split the genetic information which is required and which is not required to make a protein. So proteins are our body's molecular machines, which pretty much everything that we do in our life. Here, I want to add my voice specifically to RNA and RNA structure, how it can impact each and every cellular process in our body. For example, in humans, nearly 95% of genes are divided into exons, which are the protein coding regions, and introns, which are not protein coding regions, which will be removed by a special editing process called splicing. So when this happens, a protein knows what to express and what not to express. When I say structure, I mean how the structure is arranged and folded. If you know the structure is having a specific fold, and it can have a clear idea which elements can go and interact with it, which elements can go and bind with it. RNA has the ability to fold structures locally, and it can expose or it can hide a specific region where an interaction can happen or interaction can be hidden. So if you see here, the example that I have shown here, the first one where in an RNA, all three exons gets the signal, and there is no structure inhibition, where the exons are staying together as a happy and functions well as a family. But on the other hand, if you see sometime, when there is a structure attached with an exon, which makes the exon to be included in this process. So what we found out, the structure attached with the exon, it not only makes that exon unhappy, it also confuses the other exons. Oh, who am I? What I'm doing? Where I'm going? It's totally like undisturbs everything. The protein specifically I'm working on, it's highly expressed in brain, and it forms a very specific role in several neuronal function. So when people don't have this exon, if there is an imbalance between this kind of exons, what happens? People often tend to miss meetings, they forget places, they find it harder to think and imagine, and even for making a small movements for them, it's become a really difficult thing in their life. So what we said to find out, in addition to this, we were thinking this is happening in all the cells on a massive scale, but we still don't know which protein has which set of exons and which proteins exclude which set of exons. So we are trying to identify hundreds and thousands of exon-intron junctions, whether it is a structured, and that structure plays a decisive role for inclusion of these exons and intron. So from the initial investigation, I would say it's very clear that we found a correlation. This kind of a RNA structure can make a huge difference for breaking a structure or making a structure. Thank you so much.